pollen pistil interaction in angiosperms. Pollen pistil interaction in angiosperms is start with pollination, end with fertilization. Pollination means pollen grains are deposited on the stigma with the help of factors. That factors are biotic factors or abiotic factors. During the pollination, the pollen grains are contains 3-celled stage or 2-celled stage. 3-celled stage mean 2 cells are male gametes. They are also called sperm cells. One is tube cell, nucleus. If the 2-celled pollen grains, one cell is tube cell, another one is generative cell. The 2-celled stage or 3-celled stage pollen grains are deposited on the stigma. On this stigma, pollen grains are deposited is called pollination. In all types of angiosperms, mainly 2 types of stigmas are present. One is wet stigmas, another one is dry stigmas. Wet stigmas are always in wet condition of the stigma surface. They are contain glands on the surface of the stigma. That glands are released sugar chemical or sucrose chemical substance on the stigma. Dry stigmas are also produced sugar chemical substance after pollination. See, after pollination on this both stigmas, means on the dry stigmas or on the wet stigmas, all types of pollen grains are deposited. From that all types of pollen grains, some pollen grains are same species of the pollen grains. The same species of pollen grains germinated. They are growing a pollen tube. And other species of pollen grains degenerated or degraded or disappeared on the stigma. Same species of pollen grains growing a pollen tube. The pollen tube terminal part containing tube nucleus and 2 sperm cells. The sperm cells are also called male gametes. If the pollen tube is developed from the pollen grains, it enter into style. And in all types of angiosperms, the styles are mainly 2 types. One is halo space of styles, another one intercellular space of styles. If halo space of styles are there in angiosperms, the pollen tube easily enter into halo space and it reach to the ovule without any disturbance. In intercellular space of styles, the pollen tube enter into intercellular spaces and finally it reach to the ovule. By the halo space or intercellular space of styles, finally the pollen tube reach to the ovule. If the pollen tube reach to the ovule, it enter into, means the pollen tube enter into ovule by 3 ways. Entry of pollen tube enter into ovule 3 ways. One is porogamy, second one is chalazogamy, third one is mesogamy. Porogamy example is hibiscus. In hibiscus, the pollen tube enter into ovule through the micropylar region. Pollen tube enter into ovule through the micropylar region is called porogamy. In chalazogamy, Cucurbitaceae family. The Cucurbitaceae family of ovules, pollen tube enter into ovule through the chalazal region. pollen tube enter into ovule through the chalaja is called chalajogamy. In Casuarina plants, pollen tube enter into ovule through the integuments is called mesogamy. Pollen tube enter into ovule through the integument is called mesogamy. By these three types of entry of pollen tube into ovule. And finally, after entry of pollen tube, the pollen tube reach to the micropylar region. If the pollen tube reach to the micropylar region, it enter into embryo sac. Initially, the pollen tube enter into ovule by 3 ways, and finally, the pollen tube reach to the micropylar region of ovule. If the pollen tube reach to the micropylar region, the pollen tube is received by the synergids. The synergids are having filiform operators. The filiform operators mainly helps in receiving of pollen tube. If the pollen tube is received by the synergids and the synergids are ruptured and pollen tube enter into the synergid cell and the pollen tube terminal part is ruptured and the pollen tube terminal part containing 2 equal male gametes and the 2 equal male gametes are released into embryo sac. From the 2 male gametes, 1 male gamete fertilizes with egg cell to form diploid zygote. This fertilization is also called syngamy. It is also called true fertilization or real fertilization. It identified by Strasburger scientist. In syngamy, zygote is formed. The zygote is formed, it present means the zygote present at the micropylar region of the ovule. And the second male gamete reach to the center region of embryo sac. In center region of embryo sac, 2 polar nuclei are present. The second male gamete fertilized with 2 polar nuclei is called triple fertilization. Mean triple nuclei are fertilized. Triple means 2 polar nuclei and 1 is male gamete nuclei. The 3 nuclei are fused to form PEN, primary endosperm nucleus. The primary endosperm nucleus develop into endosperm. This fertilization is called triple fertilization. It identified by Navachin scientist. In all types of angiosperms, mainly 2 fertilizations are present. One is syngamy or true fertilization. Another one is triple fertilization. So these 2 fertilizations are present in the angiosperms. So these are also called double fertilization in angiosperms. By these double fertilization in embryo sac, diploid zygote is formed at the micropylar region. It develop into, that zygote develop into embryo and polar nuclei fertilized with second male gamete to form PEN, primary endosperm nucleus. It develop into endosperm. Endosperm is a nutrient tissues. The nutrients are supplied to the zygote and it develop into embryo. It is a pollen pistil interaction in angiosperms. The pollen pistil interaction start with pollination and end with fertilization.